Okay, well, let's get started on the clock. Thank you, everyone, for coming to the Unlocking Better Zebrafish Antibody webinar. My name is CJ from Boster Bio. We make antibodies for the past 32 years, 33 years now. And today we want to talk about how we are going to solve the antibody shortage crisis, or not as much as a crisis, it's been going on for decades, but that problem. We're going to make that problem go away, and we're going to talk about how we're going to do that today. And my co-host, Amy.
Hi there. My name is Amy Robbins. I am a scientist at InVivo Biosystems. We're a company located in Eugene, Oregon, and we use C. elegans and zebrafish for a lot of custom genetic and phenotypic modeling projects. And I'm on the zebrafish side of that. So I'm really happy to be part of this webinar and talk about some of my experiences with zebrafish antibody seeding.
Oh, lovely, lovely. Thank you very much. So first, I will talk a little bit about why zebrafish is great, and then we'll jump right into our solutions for zebrafish antibodies. And as the original promise states, we will talk a little bit about how we design antigens to make good antibodies. If anybody take further interest in that, I can talk about that all day. Just raise some questions for the Q&A. So first, something that you probably already know, but I still need to touch is why zebrafish is such a great model organism. I think the two unique qualities that make it really stands out from the rest of the call it non-model organisms is the fact that it has a reasonably size where its larvae stage up to a certain age is all completely transparent, which makes any kind of developmental study, immunostaining very easy to do. You don't have to do tissue clearing for any of the baby stages. So transparency, one of the biggest advantages that's unique to zebrafish. And its relevance to human disease is also much higher than, let's say, Drosophila or some of the less non-vertebrates. So it has it hits the sweet spot between cheap to maintain and easy to expand versus relevance to human diseases. And it has all the other traits of other non-model organisms, which is relatively easy to manipulate genetically compared to, say, a mammalian like mouse or dog or any mammals. It has high fecundity, so one passing can give you thousands of offsprings, and it develops very fast, so that just overall makes it very easy to handle compared to a more mammalian model organism. However, there's a big challenge that we all feel that we all have experienced in the past, which is there's just hard to find antibodies in most cases for any biomarker in zebrafish. And when I say that, it's really for anything that's not human or mouse, it's hard to find antibodies. But today we're talking about zebrafish specifically. Only 8% of all the proteins in the zebrafish genome has any documented antibody usage. And that is from data on zfin.org. So for the 92% of all the proteins in the genome, there are no commercially available antibodies. Now, what happens when you do run into that situation where you don't have a good antibody? There are two options. You either try to repurpose an antibody that's made for a autolog in either human, mouse, or some other species, try to get it to work, or you custom make an antibody for that protein of interest. The advantage of repurposing an existing antibody is that it's available right away. The cons are, if you have ever tried to get antibodies to work, you can totally relate to this, that these antibodies, they are not guaranteed to work on zebrafish, and then the chances of them working on zebrafish is not as high as it could be. And I think one of the most limiting factor that you will run into is that any protein that is cross-reactive from human to zebrafish or from mouse to zebrafish, they tend to be evolutionarily conserved very much. Right? So that the sequences that these antibodies target tend to be highly conserved, even between species. So that within the species, within the same family members, there are going to be a lot of conservation sequences as well. So that if you want to really differentiate a isotype of protein or a specific family member, usually that's very hard because those special sequences usually do not cross-react. So if you want to get a very high specificity antibody, usually the cross-reactive antibodies would not be able to give the level of specificity you need. Now, if you have ever tried to make a custom antibody for a protein of interest, I think the biggest hurdle is just that, well, first it's slow and then it costs a lot of money. Not until today, right? I mean, it usually costs about, say, $3,000 to $5,000 to make a polyclonal one. For a monoclonal, it can cost up to more than $10,000. So when those two options are not within the arsenal that you have access to, there's always the option to move on to the next biomarker, which may have a antibody. But unfortunately, that just leaves a lot of potential biomarkers, unturned. And if we have now access to tools that can study those biomarkers, it would make open really many doors that were previously closed, not because they are not biologically relevant, but because there's a lack of tools, right? We want to solve that problem. First solution we have for the situation number one, which is to get a antibody cross-reactive to the protein of interest, is that Boster Bio offers to validate our antibody, which there are more than 9,000 antibodies participating in this program. We can validate these antibodies on real zebrafish tissues in a region of interest of your choosing, in the application of your choosing, free of charge before you buy the antibodies. So you can go to our website and pick the antibodies that may have cross-reactivity with your protein, or simply suggest a big list of proteins that you want the antibodies for, and we will pick through our portfolio and recommend the ones that has a chance to cross-react. And the way we will then validate these antibodies in either Western blot or staining, in a condition of your choosing, and then we'll send you a professional report, show you that it works or it doesn't, and you can make a decision whether to buy those antibodies or not after you see the professional report. And this is free, completely free, no strings attached. You're not obligated to buy, even if it works. So that is our solution for the first scenario. Right? We will validate the antibodies for free before you buy. And here are some of the samples and applications available. We have embryo to young adults to full adults for Western immunoprecipitation and for immunostaining. These are the type of samples that we can validate our antibodies on. These are our well type, and we cannot receive any sample type from you. We would be picking the closest sample we have on hand to match your condition. Now, for situation number two, where you need to make a custom antibody, the solution we have here is that we can make a custom antibody for any protein in the zebrafish genome for just $600. Right, so instead of 3,000 or 5,000, it's just 600. It covers all the way from antigen design to antigen to protein synthesis to immunization and purification, all that included, just $600. And at the end, you get 100 microgram of the antibody, with additional ones available for purchase for additional amounts of money. The success rate for this program has been very high, and to our surprise, very high compared to our custom antibody programs for human and mouse proteins. After some analysis, I have some theory as to why that is. Right, there are mainly two reasons. First one is that fish protein is just more different, less homologous to the rabbit that we use as a host, and therefore generates a stronger immune response. The second reason is more of a business reason, where the human and mouse proteins have been plowed through by many antibody companies through the last couple of decades, and many of the easy-to-make targets already have antibodies made for them. So any project that comes to our hand for a human or mouse protein are usually for some tricky protein to make that are either very hard to make antibodies for, or have some tricky application they're trying to get to work. So those are by default very hard to make, right? Whereas the zebrafish is kind of the Wild West. A lot of the protein that come to our hands are, the easy to make ones. Overall, our success rate after was covering the $600 scope is more than 90%. So we're, that's overview of our solution for the custom antibody. Next, I will jump a little bit into the antigen design strategies that we employ, which is part of the reason why our success rate is high. It's not just because the proteins are easy. First, I'll go through two tips that are common knowledge that many people who have thought about immunology or have dealt with antibody production would know about. And then we'll talk about some of the kind of secret sauce that our company has that's not necessarily public knowledge. So the first tip that's common knowledge is that you need to avoid homologous sequences. So you have one protein and the parts of it is kind of shared across all family members. You don't want to use that sequence. Use the sequence that are unique. Right? This point is kind of simple. I'll just leave it at that. The second point is you need to pick the epitopes to target according to the application needs. So, for example, if you're using Western blot, right, Western blot aggressively retrieves the antigen so that everything inside of the protein is exposed to be accessible to antibody. So you can just pick any sequence that are strong. However, if you're using something like immunoprecipitation or IHC or IF, you want to ensure that the epitopes you pick are on the surface, right? So that anybody can target them. I mean, there is a reasonable amount of effort put into antigen retrieval in IHC, but not nearly as rigorous as Western blot. Right? So pick the surface epitopes is very important. And thankfully, this is becoming very easy to do now with AlphaFold predicting reasonably accurately the structure of any protein. Right? So those are two tips that are common knowledge. And here are some of the things that we do special as a company. First is that we use recombinant protein instead of peptides for immunogen. Recombinant protein is, in our experience, can generate antibodies with at least 4 to 10 times more signal in immunostaining. And that's because recombinant protein is just much larger and has better immunogenicity, and it also retains the tertiary structure of the protein. So why do some of the companies that do custom antibodies do not use recombinant protein? To my understanding, is mainly a reason, a scenario of lack of capacity, like they don't have access to a recombinant protein production line that can make recombinant proteins cost effective enough, right? Because the commercial cost of making a recombinant protein would be in the $2,000 to $3,000 range, and that could be more than the cost of the entire custom antibody project itself, right? So usually, if you don't already have a recombinant protein for immunogen and you want to engage someone to do custom antibody for you, they will recommend you to do a peptide just because of convenience. Second is we pick the right hydrophobic and hydrophilic amino acids mix so that the immunogen would have better solubility so that it can actually be expressed to begin with. And sometimes when we would have to work with a highly hydrophobic region, we would flank it with some randomized hydrophilic amino acids. That way, it kind of gives a flotation device, so to speak, so that the protein actually floats well and expresses well. The third tip is that we predict and maximize an immunogenicity based on largely local conformational contrast. I'll go into that a little bit as the last tip. This is actually one of the most important things that allows us to deal with the most difficult antigen. So I'll talk about it at the end. Oh, there are some. Okay, so first point, avoid homologous sequences. Only pick the ones that are not very same. Second is pick surface accessible epitopes, use recombinant proteins, and pick the right mix of hydrophobic and hydrophilic amino acids. The last point, right, of local contrast of amino acid side chains or amino acids is that think of the antibody and antigen interaction is often referred as to a lock and key mechanism, right? If you think about it, the CDR region of the antibody have to couple onto the antigen. So just like rock climbing, it's the easiest to grapple onto if you have something that sticks out or crevice that you can stick your finger into. It's hard to grapple onto a flat surface, right? So the local contrast between neighboring amino acids, in our experience, has turned out to be a very important deciding factor of just how easy it is for the host to generate a highly specific antibody for the antigen. There are some caveats to that. For example, when the contrast is too large, like for example, you have a bunch of very short amino acids, and then you have one like Y or W that has a big benzene loop sticking out, that would be like too much contrast. But overall, local contrast is the thing that really pushes over the edge and make a really great antibody. With that, that's the end of my part of the presentation. I'll turn it to Amy for the rest. Amy, would you like to share your own screen or
Sure. Yeah, I'll just do that so I can drive the slide progression better. So let me
yeah,
just pull
sounds
that
good.
up here.
Mm.
And is that in presenter mode for you guys? Okay. I'm going to take that as a yes, CJ, even though you're muted.
I can see it. Yeah.
So thank you, CJ, for all those technical insights into what it takes to make and design high quality antibodies. I'm going to talk more about the user perspective. And I'm from InVivo Biosystems. We are a small company located in Eugene, Oregon, and we use C. elegans and zebrafish as preclinical models for a variety of purposes. My name is Amy Robbins. I have a PhD in molecular biology from the University of Oregon, which you may be familiar with Eugene, Oregon being a major hub for zebrafish research dating back to the 1970s, and we're home to the Zebrafish International Research Center and a variety of other sort of zebrafish hubs. So InVivo Biosystems is a small company that does a lot of work. We have currently about 10 patents, 30 employees, 30% of our staff are PhDs. We've created over 4,000 genetic models, 250 customer publications, and average years in experience in genetics is 18. So we are definitely all about the genetics and phenotyping. Some of our offerings and partners, we help clients from biotechnology, pharmaceutical spaces, and academic clients. So we can do custom genetic modeling, a lot of drug discovery and toxicity testing, as well as compound efficacy testing for compounds that clients are already interested in and just want to know some more information about in an in vivo model. And just a little bit more information about the types of genetic modeling that we can do, particularly in zebrafish. Some of our offerings include precise point mutations. If you want a model that has a specific mutation to match a known disease phenotype, we can do that. We've recently been exploring a lot of different techniques for knockouts, including F0 crispants, so that we can get started right away, as opposed to waiting for the time to derive full alleles. And we also do offer knock-ins, which take longer, as people familiar with zebrafish genetics know. But we really try to work with our clients to select the right model for their research applications and then customize projects around that. I've also listed here some of our C. elegans offerings, which I'm personally less familiar with, but that we do quite a bit of work with as well. And actually, InVivo Biosystems got started as a C. elegans company, so that is also a major branch of our expertise. So now on to where we would use antibody and immunostaining in this space. Of course, there's applications for validation in genetic models. If we are testing proprietary compounds and seeing if they're efficient at rescuing phenotypes or ameliorating phenotypes, that could be another space where we're doing a lot of antibody staining, as well as disease phenotyping and toxicity testing. So all of these things get at the larger goal of understanding molecular zebrafish models. And when CJ asked me to jump in and share some of my thoughts on working in this space, immediately my brain went to all the struggles I've had through graduate school and in my current job of trying to get these dang zebrafish antibodies to work. And one story in mind just popped out to me immediately. So it is no secret in the field that finding reliable antibodies for zebrafish is a huge headache. And I thought of this experience that I had in graduate school where I had a protein of interest that I was interested in visualizing. I had a beautiful disease phenotype. I had some interesting mechanistic insights, and we were ready to publish this paper. And we really, really needed to visualize this somehow in order to take it to the next level. So because there were no antibodies available that have been published in zebrafish, other than some homemade ones that were not available for me to use, although I tried. I ended up just looking for anything that could work. I made a list of about 50 antibodies that had any kind of immunogen information available. And that also varied wildly because some of these companies will say, they might give you a range of amino acids of where the immunogen is. A few of them will give you the entire sequence. Many of them won't give you any information at all, or they might just say N terminus, C terminus, something like that. So that was very difficult because I was using protein homology to try to identify what of the published antibodies would be the best fit for my particular model. And I did end up testing 4 antibodies, but I think my threshold was like 80% homology cutoff in a conserved region. And I was able to find a usable antibody. This process took months, and I would say I was lucky because there's no guarantees that I could have found a usable antibody in that case, even if I did all of that work. And this story is not unique. I think a lot of people working in the zebrafish space have similar stories and can relate to all of this, or some of this, if not all of it. And this really cost a lot of time, labor, money, reagents. This particular disease model was a rare disease model, and we had a lot of trouble getting even enough fish to survive. So we lost valuable samples testing these reagents. And because we didn't know if we could have a successful antibody, I was in parallel looking at in situ visualization options to see what else we could do. And now in hindsight, after listening to all this incredible information about the high success rate of custom antibody offerings, we could have designed and tested an antibody as soon as we knew that we were interested in visualizing this and saved all that time and trouble, and also gotten a publication much faster. So that's just from my experience, how this could have really improved that process for me. Some other advantages of working with Boster Bio antibodies from my experience with InVivo is I don't know of another company that offers fluorophore conjugated primaries. For some reason, almost every antibody that I want to use in zebrafish seems to be from a rabbit host. So that makes multiplexing really difficult because you can't unless your primary antibody is pre-conjugated to different fluorophores. So I'm thinking of a recent project where we were interested in visualizing protein complex in the zebrafish retina, and we were able to get 2 conjugated antibodies, both from rabbit host, one conjugated to a red wavelength fluorophore and one in far red. And so we could do that at the same time and also skip the secondary antibody staining step. So that was really nifty because I've done a lot of antibody staining and I'd never heard of that before. Also, being able to visualize your tissue and validate it before you start your process, that just takes a lot of pressure off of the people performing the work on your team to have Boster Bio be able to offer that. It can save you that labor and optimization efforts, which, as many of us know, can just drag on and on. And then finally, I just want to re-emphasize the $600 price for a custom antibody is, in my mind, the price of just any off-the-shelf polyclonal antibody. It's kind of mind-blowing that that's a real offering because, as CJ pointed out, these custom orders are often in the thousands of dollars, and that's just the sort of expected price. So to be able to design and be part of the process, knowing the immunogen sequence, knowing which epitopes you're going after for the same price of getting a typical polyclonal antibody is really something special. So those are just my insights as a user, and I'm gonna, I guess, kick it back to CJ if you want to handle the Q&A, and I can jump in as appropriate.
Sounds great. Thank you very much, Amy, for sharing your experience. So for any attendees, if you have any questions, you can type in the Q&A session, and we will pick the ones that you want to answer. While you type that, I'm going to talk about a few questions that some of you have submitted ahead of time. So let me share my screen here. So the first question is about directly conjugated antibodies. I mean, it wasn't clearly phrased as to what that person wanted to know about directly conjugated antibodies. So I will just show you what's available on the site. So these common fluorophores can be obtained with just a little bit extra cost. And if you want a special conjugate, we can offer that as a special custom service as well. This is what the $600 custom antibody page looks like. You can see the program details by downloading this document, and then you can just tell us what targets you want to be made by sending us a message.
CJ, could you go back to the options for
Yeah,
conjugation?
yeah.
So this is really neat to me because not only is it offering conjugations of different fluorophores, but basically you can select the ones that are appropriate for the tools that you have. Say if you're working in a GFP reporter line, or if you only have access to microscopes with certain wavelength filters. The ability to customize to this level is not something I've seen before. So that is just made me really happy as a user.
Right. Oh, thanks for David for this question. Can you conjugate a fluorophore to any antibody in your list? So to any antibody that we have this free validation badge on, we can conjugate them custom to any fluorophore. Of course, if we don't have that fluorophore, we'll have to acquire it commercially. But in theory, it will be basically us doing a custom service if it is not already part of the classic fluorophores or some of the branded fluorophores that we have, have in stock. So yes, anything that has this list, we can do custom conjugation as well. So anything that has this option. So thank you for that question. The other questions that I received before the webinar is how to make a good specificity antibody, how to make antibodies with good specificities. So as I pointed out in slide, which one was in this slide, that the advantage of a custom-made antibody is that we can target specific sequences. That's turned out to be very helpful in some of the interviews we've had with our customers. They claim that even though they have succeeded in getting some cross-reactivity going with commercially available antibodies, the staining is not nearly as clean as the custom-made antibodies. Another important part of this in the antigen design is to avoid large regions of highly hydrophobic regions. So if you haven't never run into this before, I just know that hydrophobic interactions are about 10 times as stronger than a hydrophilic interaction, so that they're just super strong and long in the range that they can affect. So if you have a large range of highly hydrophobic regions, they just become very sticky, right? So that they will bind to just about anything. So you want to avoid like large continuous sequence of hydrophobic amino acids. That will also help with the specificity. Otherwise, you would just end up getting a very sticky protein that sticks to everywhere, or the antibody that recognizes that sticky thing will also stick to other things. So avoid large spans of hydrophobic amino acids. And I got another question here about why we do this. How can we do a custom antibody for $600? Okay. Before that, I have another question. Brian Corfold, thank you very much. Given that you're generating recombinant proteins as an antigen, doesn't that make it difficult or impossible to make phosphor-specific antibodies? So yeah, the phosphor-specific antibody in general, it's hard to make them in polyclonal because what it requires is that you would make the antibody first, and then you need to cross-absorb the antibody with like a non-phospho antibody, non-phospho antigens, and you may have like less than 10%, probably less than 5% of the antibody left. So the yield is very inefficient. The best way to make a phospho-specific antibody is to go through technologies similar to our next-gen antibody discovery platforms, where we use a very high throughput screening upstream pipeline to identify clones that are specific only to the PTM, or in this case, phospho-specific antigen. We have a special technology that we love to talk about, but maybe not in today's webinar. that can screen through the entire spleen of a rabbit in just 2 days, which we're talking about hundreds of millions of clones. And that technology allows us to identify the rare populations of B cells that produce antibody that are just specific to phospho-specific or any kind of modified antigen and not reactive to things that we don't want. Because in the high throughput screening stage, we're actually able to negatively screen against like the non-modified versions of the protein. So we have a technology like that. If you're interested, feel free to reach out to us to know more about it. It's a lot more than $600. So if you really need a post-translational modification specific antibody, totally doable. We have almost a guaranteed way to pick them out, but it's a bit more expensive than this. This $600, yeah, thank you, Brian. The $600 offer is mainly to help the community roll out the infrastructural tools across the entire genome with ease. That's our goal. We're basically doing it at cost here, right? Like we're hoping to make a bunch of good antibodies, and so that people later on will buy more of it, and we only start making some cash flow at a second antibody sale. So that's kind of our vision, to work with the community to really build this out. Yeah, because our reasoning is pretty simple. That somebody has to do it, right? And we have the ability to do it, so we do it. The free validation, why can't we do the free validation? The way we think about it is we're doing hundreds of validation every week anyway. We were just kind of prioritizing the validations that somebody has raised their hands and wanted to see. So to us, it doesn't really change the amount of work we have to do. So that's a win-win for all of us. Okay. So those are the questions that I have received before the meeting. If anyone else have any questions, feel free to put them in. We will let it go for another 3 to 4 minutes. But otherwise, thank you all for coming today. If any of you are still in the process of typing one, click the raise hand button. Otherwise, you can always send your questions and inquiries to us after the show. For any custom antibody related inquiries, please send it to services@bosterbio.com. And for any free validation, by the way, the free validation program is not just available for zebrafish, it's available for human, mouse, and rat samples as well. The email for that is freevalidations@bosterbio.com. Amy, any finishing words?
Well, I was thinking about when we were meeting and going over these slides, it might be helpful for the audience to hear about how you test on zebrafish samples, because that was one of my questions for you. Is it customers sending samples to you? Do you have samples available? What is that process like?
I see. So we have went through the trouble of kind of really learning how to handle zebrafish. We have a vendor where we can procure these fishes from. So we have on stock like pre-sectioned whole bodies of zebrafish, and we have lysates and all of that in stock. And for any kind of embryo stuff that we need to do whole mount, we just kind of acquire them fresh and prep them. These are all wild type zebrafish. So we would take the antibody so it's only our antibody using our samples, using this. So we will take our antibody that customer requests to validate on these wild type samples in the region of interest chosen by the customer, to do the experiment, whether that's Western blot or IHC or IF, or generate a professional report and send that to the customer after the validation is done. So I can kind of show you what that report looks like. Just have this is a flow. For any type of histology applications, we actually have histologists on staff that will make a comment, that will make a rating to it, and et cetera, et cetera. We'll also include details of the actual protocol we used, how much incubation, what the conditions are, so that it will give a pretty good starting point for the customer to start optimizing their own experiment. Does that answer the question? Any particular details I left out?
That's awesome. Thank you.
Well, without further ado, thank you all for joining the webinar today. We're gonna send out some coupons for the services. Oh, got another question. What species are they made in? So the antibodies are by default made with rabbit, and we have the ability to make antibodies in other animals. not for $600. Price may vary, but these are some of the animals. So we got rabbit, chicken, goat, llamas, mice, guinea pig, donkeys. Those are the main ones we have. Chicken, yeah. So we can make a custom antibodies polyclonal in all of these animals, and we can make monoclonal in mouse, rabbit, and llamas. Thank you very much. Thanks for live. And for any further questions, feel free to email them to us and Amy. Thank you very much for attending. Have a nice weekend.